So we'll get started. So, I mean, first of all, it's a huge honor to be here. I've learned so much, and our trainees have learned so much from this workshop over the years, and it's going to be a real journey into diffusion imaging, DIPY, and so many exciting areas over the coming weeks. So I'm very honored and happy to be starting this workshop off. So I'm going to speak for a little under 45 minutes. I have an alarm here that you can see, so I will not go over time on global neuroimaging studies with diffusion imaging, tractometry, and generative AI. And even though I'm presenting, this will be a tour of work done by many people, Dr. Bram Chandio, Julio Villalon, Wendy Feng, Leila Nawalsi, Connor Owens-Walton, Gaon Kim, many of you are online here, with folks from the Jahanshad Lab, and also Dr. Gary Felidis and Dr. Harislak at Indiana University, who've contributed a great deal over the years to these findings. So here's a quick overview of today's talk. It's split into sections. If you're online, you can jump to one of these sections that might interest you the most. I'm going to tell you about Enigma, which is a giant neuroimaging consortium studying around 30 brain diseases. And I'm going to talk about how you can use diffusion MRI to study brain diseases with lots of examples from Enigma. We'll talk about how microstructural brain metrics vary across the human lifespan, and that'll take us into normative modeling, a project looking at diffusion MRI in over 50,000 subjects that's led by Julio Villalon. And then a couple of technical areas on how scanning protocols affect diffusion MRI metrics. A really cool toolkit that you'll hear a great deal about on Wednesday and Thursday from Bram Chandio, bundle analytics. And then finally, some really cool AI things on generative AI, which you'll also be hearing around about from John Heitz later in the week. So the first 5 minutes is not going to be about diffusion MRI. I'll just give you a little bit of background on Enigma. So essentially, for the last 15 years, Enigma has been publishing the largest neuroimaging studies of around 15 different brain disorders. The goal is to discover factors that help or harm the brain. Everyone analyzes brain scans and genetic data from a little over 100,000 people from 45 countries. And there's around 2,000 scientists in 51 working groups who've published the largest neuroimaging studies of epilepsy, Parkinson's, ataxia, brain injury, and also in psychiatry, bipolar, major depression, schizophrenia, PTSD, and substance use, and in the developmental area, OCD, ADHD, autism, and some neurogenetic disorders, which we'll talk about later on. So analyzing this data, there's two main goals. One is to see how the disorders affect the brain. We'll focus on diffusion imaging today, but there's other methods, MRI, resting state, EEG, MEG. And also, we won't talk about this today, but Enigma has a lot of genetic projects screening the genome for markers and environmental factors that affect the brain, its development and aging, disease risk, and treatment response. So this isn't necessary for most studies. It's just a type of way of studying the brain. So if you're looking at one cohort, that's perfectly fine. But this would be good for questions that require a lot of statistical power, like discovering genetic markers that affect the brain. It's also efficient because you can use the computing technology across the world. There's some cooperative machine learning projects. And also, obviously, for team science, it's not just the data, it's the expertise of all the folks looking at this. So the data is all over the world. Here it is. It's spread out. It's not centralized. The little key at the bottom is the disease that the people study in different areas. So obviously, there's very diverse neuroimaging data spread throughout the world. And the projects are organized into, in the middle, neurology, around the edge, psychiatry. If you want to study panic disorder or social anxiety with imaging, or for that matter, there at the bottom, things like autism, there's a group for you. If you want to study brain injury, there's many different kinds in the military, in sports, in children. So there's groups dedicated to these clinical questions. And then there's technical groups that'll help you analyze data. So the diffusion MRI, very much with DIPY, there's many other types of data, both imaging and genomic, that there are technical groups focusing on. So there's sort of an overview of all the different Enigma projects. And this has been reviewed. If you want to review, there's some links here to different things that Enigma is doing. Lots of papers. There's been a special issue recently on Enigma. It's been in the news a little bit. Here's some of the folks leading projects. Some of them may be familiar to you. And we'll talk about some of the ways the data is analyzed. So these are studies of cortical thickness, the gray matter thickness, and it's showing you where people with a disorder on average have differences in cortical thickness relative to healthy people of the same age and sex. And the color is the effect size. So greater cortical thickness differences are shown in stronger red colors. And you could even get the T-shirt if you want to wear your data. So the way the data analysis is organized is if you are a scientist, which is sort of a dot here, you might be analyzing DTI or diffusion MRI, and you can work with analysts looking at other diseases, and the links are sort of collaborative links with other diseases and also other modalities. So we might want to relate diffusion imaging to resting state or other types of connectivity analysis. And so that's there. And the methods used, I mean, we're going to talk about diffusion MRI and DIPY today, but there's also workflows for functional MRI, MEG-EEG, voxel-based mapping, and cerebellar and other types of analysis that you can see there. So let's talk about structural imaging and microstructure imaging. So Enigma members might have anatomical and diffusion MRI scans. One way to analyze them, which is the simplest, we'll talk about much more elaborate ways later, is to split the brain up into regions of interest. This on structural imaging would be volumes or thicknesses of regions of interest. If you're using track-based spatial statistics or TBSS and the FSL toolkit, you might look at FA or microstructural measures in regions of interest. And then you might test associations between the brain measures and disease. So you might be interested if clinical measures or risk factors affect these brain measures. You might be looking at treatment. And each site runs a multiple regression. They see if the factor of interest affects the brain measures of interest. And then you do a meta-analysis. So you'd combine the effects across sites. If there's a lot of sites taking part, then each site's vote depends on their sample size. So let's have a look at how brain structure metrics and white matter microstructure metrics from diffusion MRI differ in neurological and psychiatric diseases. So just to be very clear, on anatomical MRI, you might be looking at hippocampal volume or something simple like that. But with DIPY, you could look at, if you're looking at regions of interest, the mean diffusivity or fractional anisotropy, or even more advanced NODI measures that Gary Zhang will talk about later in the week. So here are some disorders: psychiatric, developmental, neurological. And this is the schizophrenia group. So they've been scanning people at 33 sites, and they want to know if schizophrenia patients have different brain structure and microstructure than matched healthy controls. So they might run FreeSurfer. They'd get thickness and surface area for lots of regions of interest. They might meta-analyze whether schizophrenia affects the cortical thickness. And it does. So all over the brain, a Cohen's d of -0.5 means that the patients are about half a standard deviation away from the healthy controls. A d of 1 indicates 1 standard deviation drop, but that's a pretty big effect. And you can look at the effect at each site. These are the 30 sites or 33 sites involved. And every single cohort but one shows lower cortical thickness in schizophrenia. One of them is a little bit of an outlier, but you can see at the bottom a meta-analysis where the effect sizes of schizophrenia on that brain measure have been averaged together in a way that weights the contribution from each site. So let's just look at different diseases. This is brain structure. It's not diffusion imaging just yet, but we'll have a look. So major depression, there's cortical thinning in the limbic and frontal areas. Bipolar, quite a lot more. The color scale is the group difference here. Schizophrenia, a huge amount more, much more severe deficits than the first two disorders. This is a neurogenetic disorder, so the cortex isn't always thinner in disease. In this one, it's thicker. Epilepsy, very severe loss. OCD, autism, addiction. And so this is just the thickness of the cortex. You can see that the profile of differences in each disorder is very different in distribution and effect size. And you could compare the disorders. Here's subcortical regions in these 4 disorders. You can see that there's a sort of signature with some disorders being more severe, but also the pattern is different. So we're here to talk about diffusion imaging. So let's talk about region of interest analysis of diffusion imaging. So the Enigma DTI protocol is based on Steve Smith's TBSS, and we'll talk later about tractometry. But you essentially can look in regions of the brain at microstructural measures from DTI, diffusion kurtosis imaging, map MRI, and NODI. We use DIPY to calculate many of these advanced measures. And just as a recap, this is the first lecture in the course. I mean, most of you either are experts in this or you know this. But in the brain and in any tissue, water diffuses. If water is present, it can either be isotropic or it can be directionally constrained, or there might even be multiple fibers in a voxel. But in any case, this is borrowed from Patrick Hagman. The geometry of diffusion there on the left can be measured using a variety of indices. And because disease processes also affect this, we use some of these measures in our diffusion MRI analyses. So the diffusion signal, I mean, it's a diffusion weighted signal from the scanner, can be reconstructed into orientation density functions, which portray the directions of diffusion in each voxel. And then using a variety of mathematical reconstruction techniques, you can follow the fibers and streamlines and make bundles and tracks. So the whole course is going to discuss this in depth. Here's some beautiful tractometry data here, and you'll learn tools later in the week to analyze this. So here's DTI reconstruction. Here's a section through the brain at the ventricles, a lot of diffusion in the ventricles, which are fluid-filled spaces. And this is the tensor model of diffusion. If you use higher order harmonics, or you can use many reconstruction models, you'll see the diffusion is very, very complex. And so we're going to look at how this diffusion geometry is different in different diseases and with age. So you all know this, but a quick recap. If you use a single tensor model of diffusion or a Gaussian model of diffusion, four popular metrics to compute fractional isotropy, axial, radial, and mean diffusivity. There's pictures of those at the bottom left. They're scalar measures. They all are sensitive to slightly different aspects of diffusion. A color map can be made that portrays the principal direction of apparent diffusivity. If this is new to you, have a look at this review paper by Jeff Olger. It says in the paper, this will be immediately understood by the neuroscientist who has little exposure to DTI. So you're all experts in this, but if you want to teach someone it, that's a good review paper. So in Enigma's DTI protocol, each site, and they're listed there at the top, runs something based on track-based spatial statistics, or TBSS, developed by Steve Smith. In the Enigma DTI template, there's a skeleton of the FA. The measures are collected up using the Johns Hopkins University Atlas there at the bottom right. And like we said, after quality control, each site tests associations between white matter measures and variables of interest. And they do an inverse variance weighted meta-analysis to combine effects across sites. So this has been done for all sorts of diseases. Some of the lead analysts are shown here. And here's some of the papers looking at different diseases in Enigma if you want to refer to them. I'll just give you an example. So let's say we collected diffusion imaging at those sites. This is the schizophrenia working group. You could ask where in the atlas there are the greatest differences, for example, in fractional anisotropy. And the effect sizes are shown there in the graph in the middle. FA is lower everywhere in schizophrenia. The effect size is very similar to structural MRI. And again, a forest plot shows you the effects at each site. And you can see that pretty much every site agrees they found the same thing. And there at the bottom right is a really nice DTI paper in 4,000 people showing an effect on FA in schizophrenia. FA is lower, MD is higher, and so is RD, but not all the metrics are higher. There's a little bit of redundancy. So as you know, diffusivity measures tend to go up when FA goes down. So, people have correlated these to see how much independent signal there is. And some of the reviewers ask questions about this. They say, well, do you get bigger disease effects if the ROI is bigger? Because I don't know, maybe it's averaging signal over a bigger area. And there's some weak evidence for that. Do you get bigger disease effects if the protocol is better? So HCP has a really advanced diffusion imaging protocol. Some sites don't. The answer is not really. You get a slightly higher effect with very high SNR protocols. And the final reviewer question is often, does every site give you the same answer? And they kind of do. One site was hugely off. We won't embarrass them by saying which one it is, but they fixed it. And so they fixed the error, and then the meta-analysis was fine. So just comparing DTI measures with regional volumetric measures, the winning DTI metrics are the FA and the anterior corona radiata, the body of the corpus callosum, which are about the same as the strongest effects on anatomical volumes. A funny thing is that the medications don't seem to affect the DTI measures, at least in our study. So you can be sure it's a disease effect rather than a medication effect. The structural measures are actually affected quite a bit by medication, so that's sort of interesting. And just looking across disorders, in orange, there's schizophrenia effects on FA. In blue on the left, there's depression effects, which are about half as strong. And then in this genetic syndrome, 22q11.2 deletion syndrome, which puts you at risk for schizophrenia, the profile of differences is very different, not even correlated. So obviously the disorders have their own unique imprint. Now, meanwhile, in Japan, a consortium called Kokoro ran all the same methods in Japanese people with 4 disorders, nearly 3,000 people. And they found essentially similar effects. So this is Ryota Hashimoto who led that study. Let's look at it more carefully. So in schizophrenia, the DTI measures the rank order for disease effects is on the left. In the Japanese study, which was completely independent, it's about the same. The Enigma study was bigger, so the error bars are smaller, but it's roughly the same profile. So, like we said, just looking at microstructure, the Cohen's d is about half a standard deviation. So the patients are about half a standard deviation shifted on average relative to controls, and the FA is lower, the MD is higher. Turning to Parkinson's disease, this is a study by Conor Owens-Walton and colleagues. You would imagine that FA would be lower in Parkinson's disease, and late in the disease it is. In fact, let's look at this more carefully. The stages of Parkinson's disease are often assessed using the Hoehn and Yahr scale, stage 1 being mild, stage 5 being severe. You'll see this prototypical pattern in Parkinson's disease of lower FA, higher MD in the late stage of the disease. But this paradoxical effect of better, or we shouldn't say better, but just more higher FA earlier in the disease that we don't fully understand, which we're now looking into with the more advanced DIPY methods. If you have HIV and your disease is controlled by antiretrovirals, you might be interested if your brain microstructure is any different than healthy controls. It is, just like in late Parkinson's disease, but a little milder. There's lower FA in all of these regions that you see here and higher MD. It's associated with T cell counts, but not with viral load. There's also faster aging in people with HIV. You'll see I've put a superscript, so FATDF. So these folks are experts in diffusion MRI, and they didn't use the single tensor model. So many of you know this. You can fit multiple tensors at a given voxel to better reflect the diffusion. This approach uses a continuous mixture of tensors. There's other methods a little bit like this, where the FA of each component is weighted according to its volume fraction. And then you get a sort of corrected FA that correctly and proportionally represents each fiber in each voxel. And the TDF FA looks a little brighter than the standard FA. And you'll find associations with it more strongly because, as you can imagine, here's a study of Alzheimer's disease, because it's a more accurate depiction of diffusion with less error. The error which has nothing to do with the disease has been removed. And this FA-TDF measure links very well with Alzheimer's disease, with clinical scales, clinical dementia rating, for example, and even hippocampal volume. This is work by Gown Kim, who's on the line, working with Kathy Lawrence on autism. FA lower, MD higher, which is beginning to be a bit of a picture in all of these disorders, just as a general rule, with neurotypical individuals, mainly children and adolescents, having higher FA. But there's sort of a problem. So I've glossed over this. We're acting like every cohort finds the same answer. And when the OCD group, the obsessive compulsive disorder in Enigma, looked at FA and MD in people with OCD, they found that actually the duration of illness makes things further from normal. The age of onset being younger makes things further from normal. And also the fraction of people medicated affects the results. So if each study had published on their own, the results would look very, very different, ranging from no effect on DTI at all through to very big effects. So they sort of said, well, it's really important to model all of these confounds and things about the illness course that would influence what you would see. So we started to move in Enigma towards mega-analysis, so not meta-analysis. Mega-analysis is where you centralize all the measures from anonymized individual cases and better model the effects of sight and scanner, and obviously also medication. And you can also try generative adversarial models and other very clever deep learning models to harmonize the images across the scanner. So If you want to look at the different ways of analyzing multi-site data, this very nice review analyzes 4 ways to do it. You can, in number 1 and 2, just use the same protocol, and the summary stats are sent to you, the analyst. In number 3, people send you the individual measures they derive from the images. And in number 4, people can just send you the raw images. And we'll see that the math you can use and the stats you can use improves the more data sharing there is. So let's look at the data from each site. So if you centralize multi-site diffusion MRI data, you'll see that let's look at one region of interest. It could be the corpus callosum. You'll see that the histogram of FA within a group is very, very different from one site to another. And we know the voxel size in diffusion MRI affects FA quite a bit. Let's look at it carefully. So let's say 30 sites send you their data. This is just a histogram of the FA on the left and the MD on the bottom left. You'd like to sort of correct this data so that it's comparable. And this step is known as harmonization. There are different ways to do this mathematically, but obviously it'd be preferable if a site that has huge voxels had its measures aligned, so to speak, with the information being collected elsewhere. So the COMBAT family of methods has been designed to do this. If on the bottom left as a diffusion paper, one of the first ones applying it to neuroimaging. And just to recap, if you are getting region of interest measures from the brain at many sites, because each site is giving you data with a different histogram or a different distribution, it's a way to correct for that or adjust for that site-dependent effect. So if you're a visual person, this is a really good way of looking at it. So if you look up the top left, and this might be the FA of the corpus callosum, some sites like the one in blue might read very high for FA. Other sites might read like the yellow one, rather low. And if you want to model the age effect, I mean, this is rather chaotic because the site differences are getting in the way. There are 2 ways to deal with this. One is you assume the effect of interest is linear, which means that you use COMBAT. Or you might assume the effect of age is nonlinear, which is a variant of COMBAT called COMBAT GAM. GAM stands for Generative Additive Model. And this review by Johanna Bayer tells you all about this. And obviously you'd prefer plots that look like the one on the right, where the site and protocol effects have been adjusted for. So if you like algebra, this is the same thing written out in math. You, on the left there, have a measure, which is the cortical thickness, or in our case, the FA, at a certain site for participant J, for region of interest V. And you can estimate all of these coefficients. One is the effects of, for example, age and sex and the disease. Others are the site effect. How much does being at a certain site shift these measures or cause a difference in the variance of the residuals? And as it says at the bottom, you want to find the shift and scale parameters to match the residual distribution to a reference. It could be a high quality reference data set like HCP, or it could be the other studies. And if the batch size or the site size is small, you can use something called empirical Bayes to estimate these parameters. Here's COMBAT GAM. We won't go into the details. There's even something called COVBAT that corrects the covariance of data. But all it's really doing is fixing the mean and the variance and the moments of a distribution to match a reference site. If you're at a new site and you want to do this, you can do an F-test to see which moments of the residual distribution. Is it just the mean, or is it also the variance or the skew? Then you will fix the moments that deviate. You may only need to correct the mean for a certain site. And this paper talks about using a reference batch. So you can already imagine how you could harmonize diffusion data to a reference. When you use COMBAT in Enigma, the disease effects get better. So just as we said, modeling error with TDF makes the disease effects easier to pick up. Modeling site effects with COMBAT makes the disease effects easier to pick up. And there's a toolkit by Joachim Radoa that allows you to apply some of these methods in your own study. So let's look at abnormality measures, either in region of interest data or in track data. We'll take a look at that. So let's say you've aligned your site data across the world, and this is, I think, the size of the right thalamus. You've assembled a percentile chart for the measure of interest from many cohorts. This is nearly 40,000 people from Enigma lifespan. You could make a sort of percentile chart of what that measure looks like and its distribution in people of a certain age and sex. And the raw data looks like this. It's a bit messy, but this is just out in the Lancet Digital Health. You could make charts for, in this case, the volume of different regions. And you could say, does my patient group deviate on average from this distribution? Does an individual patient deviate? And so long as you correct it for scanner and other things at the site, you can use these references. And they're essentially normative charts. So let's say you used a normative model and you measured the z-score for your measure of interest relative to this chart of differences. You could do something called normative modeling. You could say, in my patient in this region, there seems to be a discrepancy between the healthy matched controls. And Julio Villalon did this for diffusion MRI. He collected data from many, many cohorts, parcellated the brain. In fact, on the bottom left, there's a huge number of public data sets. And he said, well, actually, you can look in the middle there at the average FA of the white matter, which is a little bit of an odd measure to average it up over a lot of regions, but let's use that. It sort of rises and then falls. But at each site, the curve would be a little different because remember the voxel size and other things about the protocol mean that the FA can be a little bit different. So if you were to use this normative model on a protocol that was different than the one used to generate it, you would have a sort of mean shift in the metrics. So this is very, very useful. You can use it to norm your DTI measures and see what's happening. FA tends to peak earlier in life, whereas RD, MD, and AD tend to have the lowest value later in life. You could sort of plot on the brain the age when these reach their extreme value. And you can, as we've said, use z-scoring to evaluate whether or not a particular individual or a particular disease deviates from this. And so I would strongly encourage you to check this out. I mean, you could, for example, in PPMI, which is a Parkinson's disease data set, you could check if the patients with Parkinson's disease have higher or lower MD. And from the result earlier from Connor Owens-Walton that it's a little unexpected what happens in Parkinson's disease. You actually see them shifted, at least earlier in the disease, on the unexpected side of the normals. So this toolkit by Alyssa Zhu in Neda Jahanshah's lab lets you make normative models for multi-site data. You can see, obviously, the rather chaotic data at the beginning gets converted into much better looking data on the right. And here's Alyssa Zhu explaining these harmonization tools as an Enigma workshop. So now for the cool stuff. So the last 15 minutes, we'll talk about super cool diffusion MRI methods. So we talked about the basic region of interest methods. Dr. Bram Chandio and also Iyad Bagari working with Neda Jahanshah, and also Wendy Feng, who's been looking at normative modeling and tractometry, have been developing really advanced workflows. These are in the DIPY toolkit. You'll learn about them later in the week. Not just for regional analysis, but for long tract analysis, and doing some of the things we've talked about, harmonization and normative modeling at different points along tracts. So this is work by Dr. Bram Chandio done during her PhD at Indiana University with Eleftherios and Jarek. Bundle analytics you'll hear about on Thursday. Bramsh will give a very detailed review of what you do to extract and analyze a long tract statistics from the brain. And just as a sort of preview of this, Wednesday will be a review of tractography algorithms. And then you'll have 2 hours of Dr. Chandio talking about bundle analytics and tractometry-based multi-site data harmonization. That will be on Thursday. So just to give a recap of the workflow, you would reconstruct your diffusion model, extract streamlines using tractography, break the brain up into bundles, and then this all will be explained later in the week. The same measures we've been talking about in regions of interest, you can imagine extracting them at different points along tracts. But then the same issues arise, such as the site differences. You'd want to sort of shift data from one site to another. And you can do a long tract statistics and assess effects of disease, modulators such as medication or age of onset and so forth. So this is very cool. It's a different way of parcellating the brain as well. I mean, if you wanted to, rather than use tract-based spatial statistics, look at mean values within these bundles, you can certainly do so. And this is being beta tested in Enigma. So here's a study where the following question was asked. Let's say you combine bundle measures from many, many sites. Do the effect sizes actually get better? Do they get stronger? Because I mean, it could be that it just gets worse and worse the more data you add. And those QQ plots in the middle are histograms of the p-values you get. In this case, I think it's bipolar disorder, or actually maybe Alzheimer's disease. And reassuringly, the effect sizes do go up as the number of sites data is added. And you can use a long track combat, harmonized tractometry, and other measures to look into this. So this is work by Leyland Nabulsi with Bram Chandio looking at bipolar disorder. So you'll remember that bipolar is associated with frontal lobe cortical thinning. Those of you that know your anatomy very well will know that the anterior cingulate is also implicated in bipolar disorder. And you can see in the middle there, or the top middle, the strong FA differences in that area. So, I mean, you could begin to build up a picture of the frontal limbic microstructural changes. There are also effects in the cerebellum. So a lot of these methods also partition and bundle fibers in the cerebellum. So that's of great interest. And Leila and colleagues have shown that effects strengthen as you add sites. So I'm not going to go into the plots at the bottom. Bramsh will explain these later. But essentially, as you go along a tract, you can ask what the p-value is of the statistical association between microstructure and a disease. And if it gets stronger or weaker as you go along the track, you can plot that. And that's what those p-value or log p-value charts are showing you on the bottom. Now, what about harmonization? So you can apply combat or a site adjustment at each point along the tracks. And you could argue, you'd say, well, isn't the site effect just uniform across the brain? I mean, maybe we don't need to estimate this at each point. So you could try different things. I mean, you could say that the site correction should depend on the diffusion measure and its value, and pull data across different regions with similar diffusion measures. It could depend on the number of fiber tracks present. So this general area of harmonization of along track data is a very active area of research. And there are already methods in DIPY that you can try out for doing this. This is very cool work by Gaon Kim with Kathy Lawrence doing a long track diffusion in autism spectrum disorder. So if you look on the top right, I mean, the region of interest approach, it's cool, but I mean, it doesn't really distinguish between what's going on in those bright red areas. But then there you are in the left arcuate fasciculus over there on the bottom left. And I mean, there's a great deal more detail, which I think is much more exciting for understanding the topography of this disorder. Conor Owens-Walton and his team have been looking at Parkinson's disease. And so, I mean, it could well be that a tract that's implicated in the disease, the spinothalamic tract here in Parkinson's, is of particular interest. And you want to know, is there a spatial preference for regions that are most affected? So he's been looking at 6 cohorts. This is very early data that's quite interesting there. And then finally, what about normative modeling? So we said that Julio Villalon has made this nice reference chart for DTI, and you can see whether an individual or a group deviates from the population. What if you wanted to compare an individual tract set with a database of normal subjects? And there's a number of techniques that have been pioneered for so-called anomaly detection in tractometry. So if you look on the left, there's sort of a scruffy bit in that tract, which either could be a mistake. I mean, maybe it's just incorrectly extracted those fibers, or it could be a patient-related anomaly. One approach that you'll see, and John Heitz will talk about this on Wednesday, uses generative AI for anomaly detection in tractometry. And those of you that are used to AI will recognize at the bottom a variational autoencoder. So we'll talk a little bit about what that is doing and how it gets rid of the spurious streamlines and has that nice result over there on the right. So if you look at a textbook, all these tracks look beautiful. I mean, they look like an anatomist has dissected them. But you'll know from running streamline analysis that you get some spurious streamlines that you'd really like to get rid of because they're just going to make a mess of the future analysis. And here are some of them. So this is looking at the arcuate fasciculus, the uncinate. I mean, we know that they're not anatomically correct. It has to do with the data quality. So one way of doing this that Bramsch and colleagues with Eleftherios developed is a clustering method that tidies up tractography. And you'll hear about this later in the week. You create a distance matrix on streamlines. It's the flip distance that Eleftherios pioneered in his early work. You use a latent space embedding method like t-SNE or UMAP for a low dimensional embedding of this data. And an outlier streamline, one that doesn't really cohere with the rest, will have a large distance from everything else. It'll sit in its own cluster. And in B there, you'll see that all of the outlier streamlines sit in the top left in that little spot. And you can use a clustering and outlier removal method. dbScan is a popular one to find that cluster and delete it. So it's really cool. And Jonathan Heitz, Legarreta Goronio will talk about this on I think on Wednesday. He has a slightly different method for streamline removal, which is very, very cool, also based on deep learning. So once you fix these bundles, you can do deep normative tractometry. So this sort of combines everything that we've talked about. This is Wendy Feng's work with many other people. You could say, well, in this metric, this distance metric that relates streamlines from one person to the population, is the shape, the overall geometry, or the microstructural profile any different in a multivariate sense from the reference population? And you can map this along the tracts in much the same way as bundle analytics does. It's very, very powerful. It's not a mass univariate approach like Duan. It uses the variational autoencoder to essentially look at the tract as a whole and see if its geometry is unusual after encoding this using a variational autoencoder. Now, I sort of skipped over this. So there's a number of groups. This is work from the UCLA group who are using a variational autoencoder to encode the normal variability of either streamlines or of bundles. You can apply this to either. So I'll just tell you roughly what this is. If you're familiar with PCA or principal component analysis, you can have a large number of variables, and you'd like to replace that large number of variables with a smaller number, the principal components that you're then going to be working with. A nonlinear analog of that or one of them is called a variational autoencoder. And you can use a deep learning network to look at your data and compress it. It's a very strange deep learning network. The input and the output are the same, but you force the data to go through a bottleneck. In other words, in the middle of the network, it's only going to be represented or encoded by a very small number of parameters, which forces the network to compress the data or keep the most relevant data. So when you use a variational autoencoder on streamlines, and when I say streamlines, it is a list of the 3D coordinates in the streamlines, there might be 10,000 points that it has to mash down using a component-based approach into a very simple representation. But this not only compacts the data and approximates it using a set of latent variables, it also leads to a probability density. So if you want to know if a streamline is unusual, it might be an outlier or it might be one from a patient, it'll assign every streamline or in a different formulation, every bundle, a score. And so you can then use this in a normative framework. You can include microstructure or you can include geometry or both. And this very cool paper by Wendy Feng just put online jointly models the white matter microstructure and the overall geometry of the streamlines using variational autoencoders. You could learn the normal variations in tracts for normative modeling, conditioned on age and sex. You could even make a video of tracts in people as they get older or with different stages of disease. And you could also detect anomalies and produce an along tract mapping of both microstructural and macrostructural anomalies. And there on the bottom right, there's sort of essentially a plot of the normal range of variation for different measures and whether an individual exceeds or deviates from that normal range of variation. And then that's plotted in red there in the middle. And this is also available in DIPY. It's compatible with other DIPY outputs from Ricoh bundles and Buan. So you can try this if you want to. So this is a lot of fun. Why would deep normative tractometry give you a different answer? than a long tract statistical mapping? Well, it can, because as we said, these variational autoencoders encode a very high dimensional representation into an embedding space. And they can tell you whether the tract is abnormal in very subtle ways that might not be evident from looking at a single point. On the other hand, looking at a single point is very intuitive. I mean, if you look at bundle analytics there on the right, you really know that that point in the data is statistically abnormal from one data set to another in regard to disease. So they're just very different mathematical formulations, and you can try them both. This isn't a brand new idea. I want to give credit to Maxime Chamberlain. I mean, this idea of using deep learning for streamline either outlier removal or abnormality detection has been around for a little while, and you could look at Maxime's paper if you would like to. So just to wrap up, we have 1 minute to go. Really, this sort of gives a flavor of what you could do with DIPY and diffusion imaging toolkits. So obviously, diffusion MRI powerfully detects disease effects on the brain. Depending on how strong the effect is that you're interested in, you might want to do multi-site. Single-site low-N analyses can lack power, and they may have unknown generalizability because many factors influence the disease effects. In the case from OCD, whether you found anything at all depends on how long. Oh, sorry about that. It depends on how long the people had been ill, when their age of onset was. So this sort of chaos in the literature of different findings often is due to either the medications or properties of the patient group. And in multi-site analyses, you can model these. So if you had sites that are either more severe at a later stage of the illness or with earlier onset, you can do a so-called meta-regression and see if those parameters influence the disease effects you're detecting. Also, harmonization tools and normative models can help you to integrate and compare data from many sites. It's the early days. I mean, you'll see examples this week of these being extended to tractometry. And I spent a lot of time on very basic measures from the diffusion tensor. There are much better diffusion measures. Gary Zhang will tell you about NODI and MapMRI and DKI. So many different cool measures. But the mathematics is separable. I mean, obviously, if you're using those measures, the same statistical framework that we've been mentioning carries over. And also have a look at generative AI. It's very cool. You can use generative AI with Midjourney and Sora and these cool tools that will make beautiful images from a text prompt. But you could also make a generative distribution for tracks, bundles, and their associated features using the same fundamental mathematics. So I would encourage you to read about variational autoencoders, or if you're a physicist, read about latent diffusion models, which are use autoencoders, but in a clever way to make new samples from a distribution. So a big thank you. I'm just the speaker today. This was work that was done and led by Dr. Bram Chandillo, Dr. Julio Villalon, Wendy Feng, who's a graduate student at USC, Leila Nabulsi, Connor Owens-Walton, and Gaon Kim, and many of you here online today. They also have been helped by the Jahanshad Lab, Yad Bagari, Alyssa Zhu, Talia Nir, and obviously the DIPY team in Indiana, Eleftherios and Yarek, have been fundamental in really creating a vision for these tools and how they're to develop in different areas of disease study. So last of all, if you're ever in Los Angeles, come and see us. It doesn't always look like that. That's a particularly nice day. But if you're coming through, come and say hello, and we'll chat about diffusion MRI. So that's it. Thanks so much for coming. Appreciate Eleftherios the invitation, and you're about to set off on an odyssey into the advanced methods for diffusion imaging. So good luck to all of you for the next week. Thanks very much.